Assalamu alaikum everyone. I hope everyone's doing well. My name is Noor Waseem and today I'll be giving you a lecture on enzymes part 1. I'll only be covering part 1. The rest of the two parts will be covered by the other tutors. All right. First of all, apologies for the recorded session. Inshallah, the session will be available soon today and I hope it really helps everyone. Inshallah. And if you have any questions, please text me on WhatsApp, email me, anything. I'll be more than happy to help you guys. And tomorrow's your TBL, so best of luck for that. Inshallah, so let's begin. It's not a very difficult lecture, but there are some high yield stuff that I will be pointing out, and make sure you do this. You're good with those in your exam, and especially before your TBL. I have added questions related from those area, and inshallah, I hope so everything goes well. Okay, so let's begin. Bismillah. So these are our objectives. We will be talking about zymogen, proenzyme, coenzyme, cofactor. There are a lot of names. You need to know the definition, enzymes and activation energy of the reactions, transition state, what it is, and how does it help in lowering the activation energy, the classes of enzymes, cofactors, and coenzymes again, and their function in catalyzed reactions, models of enzyme catalysis, and factors that affect the enzymatic reactions. Okay. Um, Pretty much what we did back in high school, and it's more like general knowledge. What are enzymes? Enzymes are proteins. They are catalysts. They are selective. They are specific. They can be recycled, and ACE endings, ASE, like pretty much for majority of the enzymes. Okay. One thing I need you to remember is that proteins can be RNA molecule. Okay, all the enzymes are proteins. Okay, but obviously not all proteins are enzymes, but all enzymes are proteins. They can be RNA molecules, and some of them are involved in RNA splicing, some of which are ribozymes and ribonucleic acid enzymes. And of course, catalysts, they help in increasing the rate of the reaction without being consumed in the reaction. Then there is selectivity, as in the enzyme knows what they are going to act and catalyze on. Okay, so they are specific for which they catalyze. They won't be catalyzing other reactions. They will be catalyzing their own reaction. For example, urease will only act on urea. Okay, it won't act on anything else. All right. Specificity, again, specificity is by the shape, which is a 3D shape, and enzymes are specific in the sense that they know what they're going and who they're going to act on. For example, trypsin acts on the carboxyl side of the amino acids lysine or arginine. So trypsin knows, oh, I need to be specific only to lysine or arginine and no other amino acids. Okay, that's specificity. Recycled, and then ACE endings. All right. Okay, so before we actually dig into the lecture, here's an example. Okay, so I want you all to listen to this carefully. It's not hard, but this will help you gather an overview of what's gonna come next. Okay, so let's say you are on a hill. Okay, and you start from here. Okay, give me a minute. This is the start point, and this is the ending point. This is the ending point, okay? And then here's a restaurant, okay? The restaurant. Now, you're here and you need to ascend all the way to the restaurant, have some food, and come all the way down. And you're at your destination, okay? So now, when you're standing here, you're looking all the way up and you're like, oh man, that's a lot to ascend. How do I do this? Obviously, you need a lot of energy while going up a slope, right? So what you do is you get all energized, you're well equipped, you're all ready to go up, and then it took time. You go slowly, like slow and steadily, you reach the restaurant. Okay, once you reach the restaurant, you have the food, and now you're more energized. Now you're energized again, but while coming down the slope, well, like going down the slope is much easier than ascending. right? So you easily come down and then you reach your destination, okay? This was an overview. Let's go ahead. Okay, this is what I was going to talk about. Okay, so this is basically a graph of a reaction which involves an firstly without a reaction without the enzyme, and secondly, when the same reaction happens with the enzyme, it shows us the progress of the reaction. Okay, now here I need you to see that these are the reactants and the free energy is zero, obviously because the reaction has in progress, and this is the product. Again, free energy is zero because the products are formed. But what is this free energy? This free energy is the amount of work you need to do, is the energy that the reactants need to do work. You remember when I told you that you need to go uphill and then here's a restaurant? So you need energy to go up, right? Okay, and then you come down and you're at your destination, as in the products are now formed. The high, you see, you guys must see like the highest peak here of free energy, and this is also another high peak of free energy. Okay, it's both. This is obviously without the enzyme, and the purple is with enzyme. So when you eat at the restaurant, you are fully energized. Okay, this state of high energy, this is called transition state. Okay, it's a very high energy state that the reactants go through, and it's kind of a state that helps the reactants progress into products. Okay, but the condition is the reactants have to pass the transition state regardless of an enzyme being used or enzyme not being used. All right, okay, so reactants, they use their energy that they have and they reach a point called the transition state. Once they reach the transition state, which is a high energy state, they form products. All right, so what exactly is free energy of activation that's written here? Now, this free energy of activation is basically the energy that the reactants need to reach the transition state. So they're able to break the reaction barrier here, right? They need energy, and now this state is also kind of a barrier. Once the reactants, they break the barrier and reach here, nothing can stop the reactants from reaching the products. Okay, and this is the highest point. Please remember, like you can get a question in the exam, what's the highest point of the graph, and you say that it's a transition state. Okay, and what is free energy of activation? It's basically sorry, it's the energy required by the reactants in order to reach the transition state, as in order to overcome the reaction barrier to form products. Once you overcome that, khalas, nothing can stop the reactants from reaching the products because they have already reached the transition state. Another thing I want you to notice here is the graph without the enzyme. Do you see the reactants need high energy, and the graph with the enzyme, the reactants need low energy? So which one is more energy feasible, or which one is more energy efficient? Obviously, the one where the enzyme is being used. When reactants need less amount of energy to reach the transition state, it's a very good reaction. It's a stable reaction because you don't need more energy, right? It's a very energy efficient process. But when you are not, when you're, sorry, when there is no enzyme, you need high amount of energy, right? And obviously, it's not like a very feasible reaction. The reaction will be slow, and it will take time to form the product. But with the enzyme, it lowers the free energy of activation. All right, and yeah, these are the two main points. One is the free energy of activation, and the other one is the transition state. Here, these are just notes you can read. Enzymes lower the free energy of activation. Like I said, look, the free energy of activation of the enzyme is lower. But if there's no enzyme, look at the free energy of activation. Look at the amount of extra energy the the reactants need when they're without the enzymes. Okay. Um, Again, at the transition state, a large number of reactant molecules reach this stage, like a large number. At the transition state, because it's a very high energy state in both these areas compared to the graphs, It is basically a stage where the substrate and the active site will be in the process of forming complexes, right? Exactly, they won't form the complex, but they're in the process of forming complexes because they're at the high energy state. There will be more collision, okay? There will be more proper orientation. And then once that happens, collision, proper orientation, process of forming enzyme substrate complex, they will reach their products, okay? Remember these points. Again, no effect on Gibbs, the delta G node of reaction, which is basically this, which is the difference in energy of reactants and products. So of course, there's no difference in both the one with the enzyme and the other without the enzyme. Okay, and this is kind of summary again. There is no difference in the free energy of the overall reaction of reactants minus energy of products. Look, free energy of activation for uncatalyzed reaction and free energy of activation for catalyzed reactions. Okay. All right. Next slide. Now we're going to talk about active site and substrate. Very easy point. Let's say this is the enzyme. Okay. And wait, let me use a marker. This is an enzyme. Okay. And the part of the enzyme where the substrate, or in other words, the reactant molecule would come and join, is basically the active site. And the reactant part, which comes and joins the enzyme or the active site, is basically known as the substrate. Okay. All information. This is basically a summary of what I said. Now, two properties of the active site allow the reaction to proceed. One is obviously the shape of the active site. If the substrate fits in exactly according to the shape of the active site, reaction proceeds. Okay, and the second one is the chemical environment inside the active site. Now, if the chemical environment, whatever the environment is, if it's feasible for the substrate to come and attach, or if it's good enough to attract the substrate, well and good, the reaction will proceed. And the specificity, again, is determined by the active site, okay? I think we've already covered this above. Catalyst, they lower the activation energy for the products to form. Remember this? They lower the activation energy for the product to form. And the second one, they increase the favorable orientation of colliding reactant molecules for product formation to be successful. Again, I'm talking about the transition state here, okay? All right, now, the next one that we have here is specificity of enzymes. Now, there are three kinds of specificity. One is absolute, the other one is group, and the other one is linkage. Now, when we're talking about absolute specificity, now the name itself says it. It means that enzyme will act on only one substrate or a single substrate and catalyze the reaction. It's going to be specific, like way more specific. It says that, oh, absolutely, I'm going to work on this kind of reaction and nothing else. And the example I've already given you above, urease catalyzes the hydrolysis of urea. Urease will only act on one type of substrate for one single substrate, and that is urea. Group. Again, we can understand from the name. In group specificity, an enzyme acts on more than one substrate, but the substrates are similar substrates. Now, a very good example is basically hexokinase acting on hexoses. Like, what are the similar substrates in hexose? For example, hexose, there is mannose, there is glucose, there is galactose. All three of them are similar substrates because they fall into the category of hexoses. So this is, again, group catalyze one type of reaction for similar substrates. Linkage. This name, link, look at the link. So it's more like that in link specificity. It catalyzes more than one type of catalyzes one type of reaction for a specific type of bond. All of them, they catalyze one type of reaction, okay? The difference that falls is in the substrate or if they are bond. Again, absolute, it acts on single substrate. Group, it acts on similar substrates, like I said here, mannose, glucose, galactose. Hexokinases acts on hexoses. Linkage, it acts only on specific types of bonds. Okay, so chymotrypsin, they catalyze the hydrolysis of peptide bonds. Like they're only going to act on the linkage or the bond, which is peptide. All right. Okay, let's go ahead. Classes of enzymes. A very high yield. Like if you are to get questions in your TBL tomorrow, I I'm I kind of pretty sure, but most probably you'll be getting from this slide. It looks very confusing, it looks overwhelming, but trust me, it's not. It's very easy. You just need to understand the concept. Okay, first of all, the classes, we have 6 classes of enzyme. We have oxidoreductases, we have transferases, we have hydrolases, lyases, isomerases, and ligases. Just to give an overview before I kind of tell you guys what this is. So talking about oxidoreductases, the name says it all. It's more like, you're talking about oxidation and reduction reactions, okay? Coming to transferases. Transferases means you are transferring something, okay? What it is, will come later. Hydrolases, there's involvement of water. Lyases, it means that something is going to be lysed or something is going to be broken down, okay? Keep in mind, in lyases, because you're breaking down something, when you lyse something, it's more like you're breaking or cutting down something. One reactant would form 2 products, okay? Isomerase, again, isomers, like it's more like the reaction that are involved in the rearrangement of groups, and the end result is an isomer, okay, of this compound. Ligases, in ligases, remember, 2 products form 1 product, okay? Like these 2 are opposite. Here, there is breakage, and in ligases, they both are combined. Both the products are combined, okay? Now, talking about oxidoreductases, remember, any kind of redox reaction, how do we know it's a redox reaction? In the exam, like one very easy tip to recognize the reaction is see if there is NAD, NADH, NAD+, FAD, NADPH. The moment you see that, oh, this is an oxidoreductases class, this is what I'm supposed to answer. Talking about transferases, like I said, it's more like transferring a group. For example, let's say, we'll talk about this. A reaction is A plus B, which gives C plus D. Now, what happens is, like it's transferring a part of A to B. Okay, and when that forms, that's good. The end result is obviously going to be C plus D, of which one of them would be B and a part of A, right? So it's more like transferring the group. You see here, you're transferring a part of A, which is basically serine here, to THF, and then in the final result, you get that compound here, okay? All right. The next one that we have is hydrolases. Now, if you could see this, bonds are broken down in the presence of water. In exam, if in a reaction you see water, okay, and you feel that, oh, it's breaking down this bond, as you can see, it forms 2NH3 and a CO2 after breakage, you are like, oh, okay, this is hydrolysis, no other answer, okay? It's very simple. You just need to get a concept. The next one is lyases. Again, breakdown of one molecule to form 2. A reaction comes in the exam, okay? And there is no water involved here. Sorry. And there is no water involved here. You are clear that, oh, because one product is formed, one reactant is forming 2 products, it has to be lyases, okay? And if one product is being broken down, there are 3 of the bonds which will be broken down, okay? And it's either going to be CC, over here we have CC, or it's going to be CN or CS. The way I remember lyases was with the mnemonic central nervous system, CNS. Okay, lyases, CNS. Remember that way, inshallah, it'll be easy. Okay, next one that we have is isomerases. Like I said, you know how isomers are like same formula, kind of mirror images of each other. So in this kind of reaction, you see that there is rearrangement of groups in such a way that the end result is an isomer. Like it's so clear that you can see that, oh, both of these are isomers. Okay, you just need to see them properly, and because there is no water involvement, and there is no transfer that you can see. There is no oxidation reduction reaction going on here. You're certain that, oh, it's isomerases. You just need to like focus on the compounds. If they look like way too similar, okay, they're isomerases. Now, ligases, again, it's opposite of ligases. And this reaction, it catalyzes formation of bonds. That is why two reactants, they form one product, okay? And the formation of bonds will be between CO or CS or CN. So the mnemonic I used for ligases was SUN, okay? Ligase is SUN, ligases was central nervous system, CNS, okay? Inshallah, it's very easy, but yes, it is high yield. Just to give you an overview, oxidoreductases, if it's a redox reaction, yes, you choose that. NAD, anything you see, NADPH, FAD, NAD+, you choose that. Transferases, you're transferring these groups. Sorry, I forgot to mention this. C, N, or P. If it's a look here, we are transferring C. Here there could be a P as well. Here there could be an N. So if it's one of them, C and P, you're choosing transferases. Hydrolysis, again, breaking down of bonds in the presence of water. Ligases, central nervous system, CNS, and breakage of bonds where one reactant forms two products. Isomerases, it has the involvement of isomers, rearranging of groups. Ligases, again, in ligases, you catalyze the formation of bonds where one reactant forms two products, okay? All right, formation of ES complex. Another easy concept, you know, like when I mentioned that this is the active site and the substrate comes and binds to it, right? So it's bound in a way that they're complex together, and this usually starts happening around the transition state. At the transition state, no. If in the exam you get a question that do enzyme substrate complex forms at the transition state, you say no. Why you say no? I'll tell you why. We'll do that in the next slide probably. Okay. Over here, what you need to remember is when there is an enzyme substrate complex here, you know, look at this picture. When there's an enzyme substrate complex here, and that now the transition state has been reached. While the enzyme substrate complex is formed, soon after you reach the transition state, what happens at the transition state? There is something called correct mutual orientation and proximity. Okay, so at the transition state, the substrate occupies active site in a way that it's fully complement to it, like fully, like exactly in the same direction. You see, it's bending from here and here because the end product is gonna be the breakage of this. Let's say it's a rod. It's gonna be the breakage of the rod. So remember, at the transition state, takeaway from this slide is there's correct mutual orientation, and we can see from here that it's bended in a way that it's totally in close proximity, and it's exactly the shape. Like, yes, enzyme substrate complex is also, like, you could say that they're exactly the shape and the fit in. Okay, but when they reach the transition state, there is so much energy that literally the product is at the verge of being formed. They literally fold themselves in the close proximity, and there is mutual orientation. Okay. The next one is lock and key hypothesis. Again, easy concept. Must have done in high school. Lock is basically the active site, and substrate is basically the key. I think this is just reading material. It's more like, you know, the active site of an enzyme is where substrate binding occurs, and that initiates a catalytic activity. It's easy, just read it. Now, the important part is this graph. You know, I told you that if in the exam you get a question that if is enzyme substrate complex formed at the transition state, you put no. Reason, I'll give you the reason now. Now, this is the transition state, right? Because it's the highest energy state. This is the transition state, or you can even write it here, transition state. Now, just notice that I told transition state is a state where enzyme substrate complex are like in the process of being formed, right? You see, there are enzyme substrate complex being formed here at this point and in here at this point. Even after passing the transition state, there are some enzyme substrate complex being formed. So this is the reason why at transition state, an enzyme substrate complex is not always formed. Please put no in the exam. If you put yes, that's a wrong answer. And the reason is because you can see that it's being formed way before not way before, like slightly before the transition state at this point, and even after the transition state. Okay. All right, and you see that the enzyme at the end it is recycled, as in it's in the exact same state as it was in the beginning of the reaction. Okay, and small quantities of enzyme is more than enough to be used in a reaction. Okay, induced fit model. Now here, induced, like by the name itself, it's more like the enzyme's active site. It undergoes a conformational change. Like once the substrate comes and bind, you can see the substrate, like it looks very much specific to this enzyme, right? But if you look closely, like there's very minor details that you need to look closely to in some of the slides. If you look closely, so this kind of curvy top is kind of like, okay, it's complementary here, but what about this? Now this kind of shape is gonna fit in here, but you know there's still gonna be some gap here, right? So what happens when the substrate comes and binds to the active site? The active site undergoes a conformational change. You can see this change is now exactly complementary to the substrate. Okay, so it's basically the active site changing the shape, and once the shape is changed, the enzyme product substrate complex forms, and then the end result is product, and boom, they go back to their original shape. Okay, you just need to read this, and a good example of induced fit model is hexokinase. All right, now zymogen. Zymogen is also a very easy concept. You can consider zymogen as the mask. Okay, let's say this is a protein. Okay, this is a protein, or let's say, sorry, this is an enzyme. Of course, enzymes are proteins, but oops, it wasn't that. Sorry for that. Enzyme. Okay, now enzyme has an active site here, right? Like all, they do have an active site here. Oops, sorry about that. This is an active site. But what is zymogen? What is the role of zymogen? Now, zymogen will kind of act like a mask on the enzyme. Okay, it is also known as zymogen or proenzyme. It will kind of cover that part of the enzyme. Okay, so for example, when a zymogen is sitting here and now the substrate is nearby, okay, so what happens? There will be a hydrolysis reaction on the zymogen so as to remove the mask so that the protein can start functioning. When the zymogen is on the protein, it's kind of a mask, right? It will not allow the enzyme to work. So once the hydrolysis of zymogen occur, the enzyme will start working because the mask is removed. And a good example of that is pepsinogen. Remember, all the gens are zymogens. Okay, so pepsinogen, it is an example of a zymogen that is produced in the pancreas, and it is kind of masking the enzyme pepsin so as to kind of protect it from protein digestion in the pancreas, and and protect it from other enzymes. All right, so it's a mask, a very easy concept. Remember it as the mask. Okay, all right. Now, factors affecting enzyme activity. Again, an easy concept, but not too easy. Factors affecting the enzyme activity, they include the environmental conditions, which are temperature, pH, ionic concentration. Please remember all three of them. Temperature is most important here. Then we have cofactors and coenzymes, and then we have enzyme inhibitors, which will be covered in your next session, which is enzymes part 2. Environmental conditions, we're going to talk about temperature. Okay, now one thing I need you guys to remember from here, which is very important, is Q10. What is Q10? It is a temperature coefficient. Okay, and it is basically an increase in the rate of reaction with a 10 degrees temperature rise. Okay, to make it easier, it says, let's say if the temperature changes from 10 to 20, temperature coefficient says what is the increase in rate of reaction when the temperature has been increased by 10 degrees, okay? When there is an increase in 10 degrees, what is the rate of reaction? This is what temperature coefficient means. But in most of the chemical reactions, the Q10 is 2 or 3. What does that signify? It means when a reaction is changing from 10 degrees Celsius to 20 degrees Celsius, it means that there is an increase of plus 10, right? This is what Q10, this is how Q10 is defined. So what is the rate of reaction? Most of the chemical reactions, they have an increase in 2 to 3 times, more like doubling or tripling of the reaction rate within this timing, okay? All right, that's what you need to remember. And yeah, you see Q10, like there's a 10 degrees increase in temperature, 10 degrees increase in temperature, that, oh, Q10 will be trying to figure out that, oh, what's the increase in rate of reaction when this has been increased by 10 degrees, and then 20 to 30, again, increase in 10 degrees. What is the increase in rate of reaction? All right, just remember that most of the reaction, it's double or tripling of the rate of reaction with an increase of 10 degrees in temperature. All right, now let's talk about the ideal temperature, or let's say the optimum temperature for enzymes, where the enzyme is at its highest activity. That falls around 35 to 40, somewhere around here, like 40, closer to 40. I think it's for some other organism maybe, but mostly even in humans it's around 35, between like 35 and 40. But at high temperatures, for example, at 70 degrees, the enzyme is obviously gonna denature. Okay, this is the reason why it's red, is because it indicates that, oh, it's high temperature. So that is why the rate of reaction falls, because the enzyme is being denatured. Okay, that's a 70 degree. Next is pH. More or less the same principle. Just remember that here the optimum pH for trypsin is around 9-ish, yeah, and for pepsin it's around 2.5. No, it's around 2. Sorry, yeah, it's around 2, and for trypsin it's around 9. Okay, so again, at extreme pH levels, what happens? There is denaturation of enzymes, similar to how there is denaturation of enzymes at high temperature. Okay, so this will cause a change in ionization. What will happen? The substrate will be unable to be binded to the active site. Factors affecting enzyme activity. Another one that we have is cofactor and coenzyme. Yeah, kind of high yield as well. You can expect questions from here. Okay, so coenzymes are non-protein molecules that help the enzyme function. Okay, let's say this is an enzyme. Okay, there is an attachment of something here. This is not a substrate though. Do not confuse this with a substrate. Substrate is the reactant that will come and attach in here. For now, we're talking about coenzyme. So you know how enzymes are proteins, but coenzymes are not proteins. That is why they are non-protein molecules that help the enzyme function. So if you don't have coenzymes, the enzyme won't work, okay? And now, coenzymes, they associate with active site of enzyme. Like for sure, like you could see in this picture, if this is the full enzyme, the coenzyme kinds of come and fits nearby or into the active site, and then the substrate comes in, and then the reaction occurs. Again, no coenzyme, no reaction. The substrate won't be able to attach. So when an enzyme is attached to a coenzyme, the name that we give is holoenzyme, which is active enzyme, because now the coenzyme is attached to the enzyme, so the enzyme is ready to function. The enzyme is ready to accept the substrate. Okay, but if there is no coenzyme and the enzyme is sitting all alone, it is an apoenzyme, and that is inactive enzyme. In the final exam, you can get a question that when an apoenzyme, it attaches with coenzyme, what does it form? It obviously forms a holoenzyme, because again, apoenzyme is an inactive enzyme. Okay, when the coenzyme comes and attaches to the apoenzyme, what happens? It starts functioning, because it is essential for the function of the enzyme. All right, here I've taken these like directly from the slides. holoenzymes can be, like I spoke, it's basically an enzyme attached to a coenzyme, right? So holoenzyme could be this, or this, okay? When an apoenzyme is bonded to a coenzyme, of course, it's holoenzyme. Like I just said, I gave an example of apoenzyme. They in the exam, look, they can say either enzyme or apoenzyme, but you need to know that when an enzyme is alone, it's called an apoenzyme, okay? Well, all right. So coenzymes could be organic molecule. They could be derived from vitamins. When an apoenzyme is tightly bonded, look, the number of bonds, it's like extremely tightly bonded to a coenzyme, it is then called a prosthetic group. That specific coenzyme that is tightly bonded to an apoenzyme is called a prosthetic group. You can read the description here. An example is FAD. Now we're going to talk about cofactors. What are cofactors? Cofactors are similar to coenzymes, but you could say that in cofactors, there are metals involved, okay? Whereas in coenzymes, we don't usually have metals involved. So cofactors are inorganic and coenzymes are also, and coenzymes are organic, okay? Please remember the examples. That example of a good coenzyme would be vitamin. Example of a coenzyme that is tightly bound would be FAD. And that specific coenzyme is called prosthetic group because it is tightly bound to that coenzyme. And then you have apoenzyme attached with a metal ion. The moment you see metal ion, you should like, oh, it's a cofactor. It's an inorganic molecule. Okay. This point, if a coenzyme transiently associates with the enzyme, it is called a co-substrate. Basically, if you have a coenzyme which is like loosely bound to the enzyme and it's almost about to break off from the enzyme, right? So it's temporary, right? It's not permanent because it's loosely bound. So it is known as a co-substrate. So here, if you want to know more about co-substrate, here is a comparison between co-substrate and prosthetic group. Like I mentioned, prosthetic group is a kind of coenzyme that is tightly bound to the apoenzyme. Co-substrate, it is a kind of coenzyme that is loosely bound. Because it's loosely bound, the name it's given is co-substrate. Okay, so think about co-substrate is after the reaction, it dissociates in altered state. Like it can't be used again. It has to be recycled by a different reaction. Example is NAD plus. Prosthetic group, again, tightly bound. After the reaction, it returns to original form because it was tightly bound and it didn't really have any alteration or changes in shape, so it came back in the original form. Whereas co-substrate was recycled, prosthetic group came back in the original form. Example is FAD plus, okay? All right. Now, quantification of enzymes. Not really high yield, but yes, they can ask you. They can give you the definition, and they're like, oh, tell me which unit may be used. So if you're converting 1 mole of substrate to product in 1 second, you use the unit called kata. If you're converting 1 micromole of substrate to product in 1 minute, you use enzyme unit. It's basically memorization. Okay, and of course, how do we quantify enzymes? We quantify enzyme based on measuring their activity. And how is that possible? How do you measure an enzyme's activity? So you look at the enzyme, a certain quantity of enzyme being converted. Wait a minute. The ability of a defined amount of enzyme to convert the enzyme substrate to product. Yes, a certain quantity of enzyme being able to convert a certain amount of enzyme into a product. That is how the activity of an enzyme is measured, and then it is quantitated. Okay. Now, isoenzymes. Here, what you need to remember is that isoenzymes, they are different forms of the same enzyme. Okay. For example, we have creatine kinase 1, we have creatine kinase 2, we have creatine kinase 3. Okay, but the thing is that they're derived from multiple genes. Okay, whereas isoforms, they are closely related gene products. They share the same biological function, but can differ in activity, regulation, expression pattern, location, other factors, often originating from a single gene. Isozymes, even though it is different from different forms of the same enzyme, they are derived from multiple genes. isoforms. They share the same biological function, but they're derived from a single gene. Okay, that's what you need to know from this slide. Talking about substrate concentrations in non-enzymatic and enzymatic. Again, I took it directly from your slide because pretty much everything was included here. Now, look at this graph. So we have the reaction velocity, and then we have the substrate concentration. In non-enzymatic reaction, as the concentration of substrate increases, the reaction velocity also increases. You see the slope going all the way up. So they are proportional to each other. But in enzymatic reactions, you see that initially they're proportional, like as the substrate concentration increases, the rate velocity or the rate of reaction also increases until it reaches a point, and now the reaction is steady. Now that specific point is known as Vmax. Now, why do we have this Vmax here? The reason we have this Vmax is because the enzyme has reached its maximum saturation where all the active site is occupied and there are no more substrates that can come and attach to the active site. So what happens? The reaction ceases or it kind of stops because all the active sites of the enzymes are used up, right? And there are no extra substrates. Even if there are, the active sites are quite saturated that they cannot accommodate more. So what happens? The reaction kind of ceases. This is basically known as the Vmax. In order to change the Vmax, remember, you can alter the concentration of the enzyme. Then Vmax will alter. For example, like I said here, the active site will be saturated. All of them will be saturated. So if you alter, let's say you increase the concentration of the enzyme, what will happen? Now, more active sites are available and more substrates could come and attach to those active sites. And so the reaction could further increase. Again, until it reaches a point called Vmax, where the more enzymes that you introduced are getting saturated at this point. Okay, easy? I hope so. That's it. I hope it was a simple lecture. And if you have any questions, please feel free to text me. Feel free to reach me via email. But there are some questions, inshallah. So these questions will be easy, okay? Because I've explained everything to you guys. And these are the high yield questions that I think even I got these in the exam 2 years back. Okay, so let's begin. The first question says, so I want you guys to read the question and then pause. And once you have the answer in mind, then play back the recording so you can understand the explanation and you can know the answer. Kinases are a class of enzymes that incorporate phosphate onto their substrates. The catalytic activity of kinases classifies them as a member of which of the following families, enzyme families. Now it's talking that a phosphate group has been added to their substrates, phosphate, right? So it includes phosphorus. So if you remember, please pause the video here. I'm going to tell the answer. So the answer is obviously going to be transferases, because transferases is basically the transfer of either carbon, I think nitrogen if I'm not wrong, and phosphorus. Okay, so this, when you're incorporating phosphate, remember phosphate has phosphorus, and this is definitely going to fall under transferase. It cannot be hydrolysis because they did not mention water. There cannot be isomerases because there is no diagram. They haven't mentioned anything like enantiomers or isomers or anything. Cannot be ligases. Okay, so cannot be oxidative reductases because no redox reaction has been mentioned here, or no NAD, FAD, nothing has been mentioned. There you go. The answer is transferases. So kinase. I know you. I'm not sure if you guys have taken this enzyme, but please don't panic in the exam if you see this name. That's just given to give you an example of an enzyme that incorporates phosphate onto their substrates. Okay, so don't worry about it. You can read the explanation here. And the next one, enzymes are efficient catalysts because they can do which of the following? A says that it can catalyze reactions that will otherwise would not occur. No. Why would it catalyze reactions that are not going to occur? Second one is decrease the free energy of the activation of reactants. Maybe we can think about it. Decrease the standard free energy of reactions? No, the standard free energy of the reaction in of an enzyme catalyzed reaction does not change. Why will it decrease? Prevent the conversion of product to substrate? No, the substrate is converted to product. It cannot convert the product to substrate. Shift the equilibrium of reactions towards more complete conversion to product? No. So the answer is obviously B. Okay, I just told you that when an enzyme is involved, it helps in decreasing the free energy of the reactant so that, the reactants don't really have to do much work, and then, they reach the transition state, and then they reach the formation of products. All right, here, this is the explanation. Okay, which classes of enzymes do dehydrogenases, oxidases, peroxidases, oxygenases, or reductases belong to? The name says it all. Uh, Obviously oxidoreductases. Okay, it cannot be ligase, it cannot be lyase, it cannot be hydrolase. Like you need to identify the name. Dehydrogenases, remover of hydrogen, peroxidases, oxygenases, reductases, reducing. So anything that's related to redox reaction, or you feel that this class of enzyme falls under the redox reaction, this is a very simple, like a very straightforward question they could ask, and the answer is obviously oxidoreductases. Okay, then here you go. Again, I have added this diagram because, of course, it's a very high yield diagram. You can see here the example that was given in our lecture was lactate dehydrogenase. So yes, we do see one of the dehydrogenase mentioned. Okay, and other than you find anything called NAD, FAD, NADPH, NADH in the question, choose oxidoreductases. Okay, which of the following enzymes catalyze rearrangement of atomic groupings without altering molecular weight or number of atoms? It's very simple. Like they've given the answer already. Rearrangement of atomic groupings. Remember when I was teaching about isomerases, I told it's more like rearrangement of groups, and then it's at the end result is the formation of isomers. So again, it's isomerases. It cannot be transferases because there is no transfer of carbon, nitrogen, or phosphate. Cannot be hydrolases because there is no water involved. Cannot be ligases because most probably they might give a picture where one reactant is broken down to two products. Okay. Yes, the answer is isomerase. Okay, now apoenzyme, when bound to a coenzyme, forms what? It's a very simple, easy question, but some students can get confused because they might be like, oh, why is it apoenzyme and why it is not enzyme alone? Like I said, apoenzyme is inactive enzyme. And enzyme only gets activated when a coenzyme attaches, right? So if they put apoenzyme or enzyme in this case, it's the same thing because both of them are inactive unless a coenzyme comes and attaches to them. They're gonna form a holo enzyme. All right, it was a very simple question, not prosthetic group because they're not tightly bound, not cofactor, because there is no, like nothing about the metal ions is mentioned. So why is it going to be cofactors? There you go. I've attached this from my slide. Look, enzyme alone is going to be apoenzyme, but when a coenzyme attaches, it will become a holoenzyme. Next, to change the Vmax in an enzyme catalyzed reaction, what do? Do you increase the duration of reaction? I don't think so. Do you decrease the duration of reaction? No. Do you remove the enzyme? No. It's an enzyme catalyzed reaction. Why would you remove the enzyme to change the Vmax? Do you alter the concentration of the enzyme? Yes, that's the answer. You alter the concentration of the enzyme. You can either increase the concentration of the enzyme, you can either decrease the concentration of the enzyme. In case you increase the concentration of the enzyme, now there will be more active sites available for the substrates to come and attach to, and so the reaction can proceed further until it reaches a Vmax again. Because again, the enzyme that you introduced is gonna have its active site saturated. Okay, all right. Thank you so much, guys. I hope it is a helpful session. And best of luck for your TBL tomorrow. Again, I'm mentioning it again and again. Since it's a recorded session, please feel free to reach out to me. You can use my number, you can use my email. Any kind of doubt regarding this lecture, you guys can text me, and I'll be more than happy to help. And yeah, here's a feedback form. Do let me know how I did. And thank you so much for listening to my recording. And I wish you guys score really good in tomorrow's TBL. Inshallah, inshallah, inshallah. Take care, guys.